This session is called Strategies for ASO Design and Optimization, Part One. Like Emma said, we'll try and schedule a second follow-up session next week to expand on this. So our goals for today are to review the different types of ASOs, to build your ASO design toolbox. So all of the different tools that we use to help us with the different parts of ASO design. We're going to walk through an exon skipping ASO case example. This is actually one of the SMC patients that we're working on. And then a takeaway should be learning some of the best practices for ASO design. And then before the next session, if you would like to, we'll give you a couple of cases that you can practice on your own, and we can review them together next time. Please feel free to stop us at any point if you have questions as we go. Don't worry about interrupting. We can kind of have this as a discussion. So I'll start by going through the different types of ASOs. So there are splice modulating, exon skipping, exon inclusion, gene knockdown, allele-specific knockdown, and upregulation or tango ASOs. I'll walk through some examples of each of these before we get into the ASO design portion. If you'd like to learn more about evaluating your cases for the different types of ASOs, we put out this paper a few months ago with the N of 1 collaborative. So feel free to go and review that. It is a very kind of complicated workflow, but it walks through all of the different criteria that kind of puts you into each of these buckets for the different types of ASOs when you're assessing a case. And if you read this and have questions, we're happy to discuss those at the next session as well. So splice modulating ASOs, an example of this is atopexin, which is an ASO that we have in a trial right now for a patient with ataxia telangiectasia. But there are a bunch of different types of mutations that can be targeted with splice modulating ASOs. When we're evaluating to see if a variant is amenable to a splice modulating ASO, we're focusing on the damage to the canonical splice junctions or coding function, the type of missplicing, if it's a gain or loss of a canonical splice site. And we're using those two together to determine how likely it is to be ASO amenable. So we categorize them as probably or possibly amenable. And then we would confirm with RNA sequencing if like to see what the actual splice impact is aside from our predictions. So these can be examples like a synonymous mutation that's creating a new splice site within an exon, and a deep intronic mutation that's creating or strengthening a cryptic splice site in an intron, an intronic insertion, or sometimes there's synonymous or benign missense mutations that cause exon skipping, missense mutations that create a new splice site in an exon, or near intronic mutations that are disrupting or weakening a canonical splice site, or like extending an exon into the intronic region. So there are a bunch of different ones that fall into this group. The next is exon inclusion ASOs. So the most well-known for this category is Spinraza, which is the SMN2 ASO for spinal muscular atrophy. So in this case, the SMN2 transcript has an exon that is always skipped, and they use an ASO to include this exon in the transcript. So these target like regulatory motifs around the exon to get the spliceosome to recognize the exon and include it to make a full protein. Gene knockdown ASOs. So this meeting will focus primarily on exon skipping ASOs, which are similar to the other splice modulating ones I just talked about. Gene knockdown ASOs work differently in that they have a different structure and they inhibit RNase H to degrade a protein. So the ASO will bind to the mRNA and it will cause it to be degraded. So knocking down the gene. Tominersen for Huntington's disease is an example of one of these. You can also have allele-specific knockdown ASOs, so it works the same as gene knockdown, except it targets a SNP on just one of the alleles while leaving the other one untouched. So again, it uses RNase H to knock down the mutated mRNA, but doesn't touch the wild type copy. And tofersen, which targets SOD1 for ALS, is an example of an allele-specific knockdown ASO. And then TANGO or upregulation ASOs work by targeting missplicing patterns or inefficiencies that happen in the wild type copy of the gene. So sometimes genes will have a poison exon that's sometimes being spliced into in some isoforms that leads to nonsense mediated decay. So it's finding those non-functional copies and targeting them with an ASO to promote more wild type protein. And these would just be in cases of haploinsufficiency, targeting the wild type allele, where even if the ASO binds to the mutant allele, it doesn't really do anything. There's just non-functional protein on that side, but it's upregulating and making more of the wild type copy. Okay, any questions about any of those before we switch to exon skipping? Okay, so exon skipping ASOs are used to skip an exon either to restore the frame of a protein in a case where an exon is deleted in the patient, or sometimes in a large gene like DMD, if there are damaging missense mutations, nonsense mutations, loss of, or frame shift mutations, like any loss of function mutation in an exon, if that exon is in frame with the coding sequence, you can skip the exon and the loss of that small piece of the protein, that one exon, can still be therapeutically beneficial to the patient. So it's still an improvement sometimes to have one exon skipped rather than having a loss of function mutation in the middle of the gene. So there are actually a few examples of these for DMD that are already in clinical trials. There's one for exon 51, two for exon 53, and one for exon 45. For our patient, we're focusing on exon 50. So we have a patient who has a deletion of exon 51. And these next slides, Heejin put together for us while she was designing these ASOs. So this is the slide that Professor Jihoon Lee shared with us. SMC9 is the patient number, but this patient has a deletion that encompasses exon 51 of the DMD gene. So these are the breakpoints in the introns. And it comes pretty close to exon 50. You can see over here. So Jihoon confirmed the breakpoints of the deletion. This is in the UCSC genome browser. And these are the coordinates of the deletion that we're looking at. So one of the tools that we wanted to highlight here is the UCSC genome browser. You can use it to visualize genomic coordinates. It has a ton of other tools and capabilities built into it. If you haven't explored it before, I highly recommend like looking up a gene of interest and just kind of playing around with it because there's a lot that it can do. It's a really powerful tool. So now we know what we're looking at is this exon 51 deletion. For some of the tools that we're going to talk about today, they require that you have the variant name written in different notation. So there's this list of rules called HGVS nomenclature, and it is a list of like all of the different ways that you can write a genetic variant. So Jihoon had given us the gDNA coordinates, which is like where the location of the mutation in the genome. For some of the tools that Heejin ran, she needed the cDNA coordinates, which is in the DMD coding region, the coordinate for the mutation. So a tool that you can use if you want to convert between different types of variant nomenclature is called Utilizer. And you can just put in the genomic coordinate and it will give you out for each of the different transcripts of the gene, the different cDNA coordinates. It's one of those things that is occasionally really helpful to have. And when you need to do it, it's good to know where to go. Okay, so for this case, we decided that our goal was to design an exon skipping ASO. So currently, our patient doesn't have exon 51. So, pending RNA-seq confirmation, our hypothesis is that the patient has exon 49 splicing into exon 50 and then splicing from there into exon 52, which would create an out of frame protein with a stop codon. Um, so we are planning to tile exon 50 with ASOs so that we're now skipping both of these exons, which would restore the frame. There is an example of someone who has designed an exon skipping ASO in Japan. It looks like it's in a phase 1/2 clinical trial there, but I don't believe they've published the sequence of it yet. So while exon 50 ASOs will be beneficial or could be beneficial for our patient who has this exon 51 deletion, they could also be used, like I mentioned previously, for other patients who have loss of function mutations in exon 50 as well, if used in conjunction with another ASO, like the one that was available to skip exon 51. So if you would like to visualize an ASO or a gene transcript, there's a tool called ExonVis that our collaborators in the Netherlands made. So this just shows you for each exon which part of the codon the splice junction is in. So if an exon is completely in frame, like it's a multiple of 3 base pairs, it will be a square like this. But if it has like the first base of the next codon, it looks like this. If it ends with like the first 2 bases of the next codon, it looks like this. So we're looking for skipping opportunities where we're creating junctions that will fit together. So like you can see up here as an example, if we were to skip exon 6, like exon 5 and 7 don't fit together. So just looking at, like, it kind of helps to look at these shapes and see which exons you could potentially skip and still have an in-frame protein. Okay, so when designing exon skipping ASOs, Heejin focused on these 3 strategies. One is the splice acceptor site. So the beginning of exon 50, the intron 49 exon 50 boundary, the splice donor site, which is the end of exon 50 and start of intron 50 boundary, and then exonic splicing enhancers or ESC rich regions, which we predict are somewhere in the middle of exon 50. So all 3 of these approaches can work. We figured we'd kind of spread our designs across them all. If you wanted to start by just working on one and testing a limited number of ASOs, you could. In theory, all of these should be efficacious in some way. So since we know where the start and end of the exon are, those are pretty simple targets for us, but to find these exonic splice enhancer regions as ASO targets, there are a few different tools that we can use to look for those. So one of them is called Human Splicing Finder. This is just what the web page looks like when you're in the sequence analysis tool. You input your variant in the transcript ID, and then it will bring you to this browser, which has a lot of different tools built in. For the purpose of today, we're just looking for the exonic splice enhancer and silencer motifs. But this is a very powerful tool if you're looking for splice predictions for other variants. So for like the splice modulating ASOs that we talked about earlier, if you want to impact or look at the impact of a variant, it does like have splice site predictions up here, MaxEnt scan, which are splice site scores. Like the higher this is, the stronger a splice signal is. And then these tiled regions are the ESCs, which are the exonic splice enhancers. So regions that help the spliceosome recognize that this is an exonic region. And then down here are the exonic splice silencers, which can do the opposite and weaken splicing of a site. There's also a branch point prediction, which is an important base for the spliceosome to bind to. It's usually an A. It's usually like 30 to 60 base pairs upstream of your exon. But this will show you like where potential branch points are. And then the GC profile, so how GC rich the region you're looking at is. So at the top here, these bases in this gray box are the exon 50 sequence. And we are looking for regions that have a lot of exonic splice enhancers because these are what makes everything recognize that this region is exonic. And we want to kind of mask those with our ASOs so that it will be treated like an intron or spliced out. So this graph in the middle here with these peaks shows kind of like the ratio of splice enhancers to splice silencers in a region. So parts where the line dips down, there are more silencers, and where the line has these peaks, there are more enhancers. So since we're trying to focus on targeting the enhancers today, we're going to look at these regions with the peaks, or the regions that have more enhancers and less silencers overlapping. So Heejin focused in on these three peaks where there were no splice silencers overlapping with the splice enhancers in the region. And yeah, so focus on areas where there were no overlap first. Another tool that you can use to look for exonic splice enhancers is called ESE Finder. In this one, you just submit your sequence in like a FASTA format. So you would have to get the sequence of exon 50 and paste it into this tool with the transcript. And it will prioritize some of the top candidates, like the strongest exonic splice enhancer regions that it's found in the sequence you've provided. So in this case, it gave the top 4 candidates with the genomic coordinates. And to be effective, the ASO should fully lap the highest scoring motifs. Okay, so this is just to recap again our strategy for the ASO design for the exon skipping ASOs. So we'd like to mask the splice signals of exon 50, so it's skipped, connecting exon 49 to 52. And we can focus on both of the canonical splice sites as well as these ESC regions that we identified. Before we move on to ASO design, I'm going to walk through a couple of like ASO best practices. These are what the field has put together kind of as like taking a bigger picture look at ASOs that have been screened and that have worked over time, and looking for patterns in the sequences. So it's generally recommended that the GC content for your ASO be between 40 and 60%. This is how you can calculate that. It's the number of G's and C's over the number of L bases. You should avoid repetitive nucleotide sequences. The more repetitive the region is that you're targeting within ASO, the more likely that your ASO is going to bind multiple times in the genome, or even multiple times like in a region in a so we're trying to have as specific of a sequence as possible. So it's really just binding to our target. The suggested melting temperature range is between 40 and 60 degrees Celsius. This is how you can calculate that. And then the length should be between 18 and 23 nucleotides. Some people have designed ASOs that are even shorter than this, and you could do that and try those as well. It's kind of a balance where the shorter ASO is, usually the less specific the sequence is, and it binds to more places off target, but shorter ASOs have been found to have better cellular uptake. So we kind of screen a range of different lengths and try and optimize for what seems like it works best for our target. And then your ASO shouldn't have any secondary structure, so it doesn't like self-dimerize to itself. And like I mentioned before, you want to eliminate off-target effects. So you're really looking for an ASO that's as specific as possible to your target sequence. And here is an article that Tim put out with a group from the N of One Collaborative a couple of years ago. That's kind of a consensus of guidelines for ASO design. And then just highlighting another tool here to add to your toolbox is the Thermo Fisher primer analyzer tool. And this, you can just paste your ASO sequence in to see if it dimerizes to itself. So kind of the same way that you would when designing PCR primers. And this will also give you the melting temperature and GC content, so you wouldn't have to calculate them on your own. Okay, so the next tool I'm going to show you is benchling. I'm not sure if people have used this before, but this is what we primarily use to design ASOs. We're still at the point in the lab where for a lot of projects, especially these splice modulating projects, we're designing our ASOs manually. So benchling we found to be the easiest tool to do this. And I can give you guys a tutorial kind of walking through how we use it. So anyone can create a benchling account. It's free to do so. And it's just another version of like a genome browser that has a lot of interactive features built in. So once you have an account, you can start by importing a DNA sequence. So you can either paste a sequence in yourself. You can also use this for like plasmids, CRISPR. There's like a bunch of different tools built in that I encourage you to play around with and see if any of them help with other aspects of your work. But for this, we usually import a DNA sequence. Like I said, you can either like upload or paste the sequence in, but it also links out to external databases where you can search for the gene itself. So here we're looking for DMD. It's going to pull it in from Ensemble for us. And you just have to specify that you want human. It's looking at HG38, the genome assembly. And then we'll search for it. And it will pull up the Ensemble transcript. So we'll just confirm that this is the transcript that we want. You can either import the gDNA sequence, including the introns, or just the cDNA sequence for different projects, like different ones are useful. I already have the gDNA pulled up, so I'll pull up the cDNA just to show you what it looks like. So we'll just take a couple seconds to load. Okay, so here we have our DMD cDNA sequence. So it just has all of the exons connected to each other, and they're all nicely annotated here for us in different colors. It has the sequence above. It has like the sense strand on the top and the antisense strand below. And then it also has the amino acid sequence for us here. You can see in these colors, and it has the protein locations as well. So if you click this little features flag up here, it will show you all of the annotations that are included on the view that you're looking at. So I'll scroll down to exon 51, which is where our deletion is. And I just wanted to show you guys that one of the features that it has is that you can edit the sequence and kind of see what the outcome is. So if your patient has like a point mutation or a small indel, you can go in and find that position and you can make the modification here, and it will show you how the protein structure changes with the mutation. So in this case, if we were to delete exon 51 and now have exon 50 connected to exon 52, we can see that all of a sudden there's all these stars appearing, which are stop codons. So this kind of confirms what we thought to be happening based on how exon viz showed us that it would be out of frame, that if we're splicing from exon 50 into exon 52, there's a stop codon here. But if we were to use an ASO to also skip exon 50, it now restores the frame, and we're not seeing any stars or stop codons in the following exons after that. So I think that this is helpful to kind of do for peace of mind to yourself, to like reassure that you're on the right track with your thinking. So this version is the gDNA. So if you had gone through that same process and just selected genomic instead of cDNA, you would get something like this, which is much longer because it includes all of the intronic sequences as well. And since we want our ASOs to target the splice sites of exon 50, we're looking at like intron exon boundaries. So we want to have them kind of overlapping with the introns as well. So I'll go down to exon 50. And you can see that Heejin has tiled the ASOs here already. So exon 50 is a pretty small exon. It is this blue region here. So yeah, it's 109 base pairs. So at the bottom here, whenever you're highlighting sequence and benchling, it'll automatically calculate for you the GC content, the melting temperature, the length of the region that you're selecting. So that's one of the reasons that we find it to be really helpful for ASO design. So she did something that we call micro walking across the splice donor and acceptor sites. So here at the splice acceptor site, at the intron 49 exon 50 boundary, it looks like every 3 base pairs, she just took a step and designed another ASO and started with just 20 mers. So each of these ASOs is just 20 nucleotides, and then you shift over 3 base pairs, and there's another one, and just walked them across this region, spanning exon or intron 49 and exon 50. And it looks like she made like 6 of them here. And then in the region in the middle of the exon where she had found those exonic splice enhancer signals, again, just walking every few bases. It looks like these ones are between 18 and 21 nucleotides. Again, they can be a range of lengths. You're really trying to optimize for GC content as much as you can as you go here. So if there's like a pretty AT rich region, you might want to make your ASO a little bit longer. So as you're kind of walking through a region, you can select them and be looking down here and see what the GC content and melting temperature look like. And then she did the same thing over here on the splice donor site. So to add these annotations for the ASOs, once you select the sequence and say, okay, I think I want to put an ASO here, you can just right click. Is that right? No, okay. You could go up here and then maybe I don't have the ability to annotate Heejin's slide. Is that why it's not working? Okay, I'll do it over here. So if you select and then right click, you should be able to create an annotation. And then you can just name it here. So you give your ASO whatever name you want. If you want to like keep them all in the same color for yourself to track them, or if you want different colors to mean different things, it's totally up to you. If you want to add any notes about your ASO sequence here for your future self, you can do that as well. And then you hit save and it will appear on the track here. So as you're designing your ASOs, we usually keep like an Excel spreadsheet going where we're keeping track of all the ASOs we're designing. So once you've designed an ASO, you just right click on it and you copy the reverse complement sequence. And then you would paste that into your spreadsheet, and that would be the sequence for ASO 1 here. Does anyone have any questions on benchling? Cool. Okay.
Not yet. Yes, I should use transferring.
Yeah, we really like it. Okay, yeah, so then here's just a recap of the ASOs that Heejin had designed for exon 50 from benchling. If you want to refer back to the slides later, we have screenshots of it included. So then the next step, once we have all of our ASOs and we've copied the sequences for them, after we've designed them, we run them through this tool called GG Genome to look for off targets. So this is a BLAST-like tool. There are others that you can use as well, but this is like a really nice simplified interface. So we really like using it for this purpose. UCSC Genome Browser has a tool called BLAT that's built in as well that you could also use, but I believe BLAT doesn't always work if your sequence is less than 20 nucleotides. So for these shorter ASO sequences, we found that GG Genome works really well for us. And you can also input a number here to see, like if you were to just run the sequence like this with the 0 here, it will only show you places in the genome where the ASO matches perfectly, but you can look as well for regions with like 1 or 2 tolerated mismatches. Or if there's like a gap in the sequence, but the rest of it matches, it can return those for you as well. So that's just something to think about when you're looking at the specificity of your ASO. Sometimes an ASO will still bind if 1 or 2 bases doesn't match the sequence. So it's possible there could be some levels of off-target binding if there are matches that have 1 mismatch. So we usually, for our first round, we'll start by prioritizing the ones that have 0 off targets. But depending on your region, you can kind of see what the results look like with up to 2 mismatches for your ASOs, and then kind of prioritize the ones that have the least number of off targets. So here are the ASOs, those 16 ASOs that we had seen in benchling that Heejin designed. And this is the type of summary table that we would put together that I was talking about. So she has her ASO name. She was keeping track of the different targets. So she had like named her splice acceptor and splice donor. That's up to you if you want to keep notes like this as well. And then the ASO sequence, as well as the GC content, length, melting temperature, and the start and end coordinates of the ASO. And then as she was running the off targets, she was looking to see how many results came back. So if GG Genome just returned 1 output that was in DMD, then there were 0 other hits. Then she would say there were 0 off targets. And then if it matched multiple places in the gene, like this one, it looks like had 4 other hits. She has 4 there. So out of these 16 ASOs, we decided to just prioritize the ones that had 0 off targets because she had a bunch of them. So for the first round of screening, just knowing that all of our ASOs are specific to DMD exon 50. Like I said, some regions you'll find that it's really difficult to find a sequence that's totally specific. You can take a closer look at these off-target hits to kind of help yourself prioritize. Like if it has an off-target hit, but it's just in an intergenic region, maybe we would be less concerned than if you clicked into the off-target hit and it was in an exon of another gene, or even in an intron of another gene. We would like to try and avoid, especially genes that are associated with disease. So that's kind of like a manual level review that you're doing of your ASOs as you go. So for now, we had prioritized these 9 that are highlighted in pink here. And GG Genome, we could walk through an example later, or I encourage you to try on your own. When it gives you the output with the off targets, it has the coordinates of every match in the genome. And if you click on them, it will link you out to the UCSC genome browser so you can see what those genomic coordinates translate to. So that's how you can check if the off target is in a gene or not. It'll just open up another window for you and it will show you. And then I just summarized all the tools that I talked about today on this slide as well. So this is your ASO design toolbox. It kind of includes all these ones that we went through today that can be helpful at different points in this process. And these same tools can be applied for designing all of the different types of ASOs, not just for exon skipping. So here are the 2 practice cases that we wanted to give to the group to try before next week. And then you can bring your questions to us, or if you want to keep detailed notes like Heejin did here and take screenshots of every tool that you used and walk us through your example and ask questions as you go or show us what you did. We'd also invite that if anyone wants to give a short presentation of one of them next week, you can just let us know. So these are two cases from Samsung Medical Center that I believe Dr. Soali has cell lines for. It's a patient with an exon 2 duplication and an exon 43 duplication in DMD. So for exon duplications, we'd follow the same principle of designing exon skipping ASOs. So for exon 2 duplication, we are predicting, but pending RNA-seq confirmation, that the duplicated exon 2 is splicing exon 1 to exon 2 to exon 2 again to exon 3. So we would design an exon skipping ASO specific to exon 2, knowing that it could bind to both copies of exon 2 on a transcript, but that some of the time it will bind to just the first one or just the second one and create some of these normal transcripts. So you would follow the same design principles that we walked through today for exon skipping specific to exon 2, and it could result in both of these outcomes. And then exon 43 is the same thing, where it's creating out of frame with a stop codon in exon 43, and you could skip it with an ASO to restore normal splicing. So yeah, we encourage you guys to give this a shot. You can also reach out to us during the week if you have questions or trouble with any of the tools. Emma and Heejin and I are happy to try and help you as you go. And then next time, we'll focus the session more specifically on gene knockdown and allele-specific knockdown ASOs, because those are a little bit different in structure, and we can talk through that a little bit more then. Yeah, thank you for your attention. And if you have any questions that come up this week while you're trying it out, you can reach out to us on Slack. We have a Slack channel that we don't really use for this project. And if you're not in it, would like to be, let me know. Or
Yeah.
you can email us. We're happy to answer your questions there. But thank you.